Chapter number 11, Transport in Plants. Have you ever wondered how water reaches the top of tall trees, or for that matter, how and why substances move from one cell to the other? Whether all substances move in a similar way, in the same direction, and whether metabolic energy is required for moving substances. Plants need to move molecules over very long distances, much more than animals do. They also do not have a circulatory system in place. Water taken up by the roots has to reach all parts of the plant up to the very tip of the growing stem. The photosynthetes or food synthesized by the leaves have also to be moved all parts including the root tips embedded deep inside the soil. Movement across short distances, say within the cell, across the membranes and from cell to cell within the tissue has also to take place. To understand some of the transport processes that takes place in plants, one would have to recollect one's basic knowledge about the structure of the cell and how the anatomy of the plant body. We also need to revisit our understanding of diffusion, besides gaining some knowledge about chemical potential and ions. When we talk of the movement of substances, we need first to define what kind of movement we are talking about and also what substances we are looking at. In a flowering plant, the substances that would need to be transported are water, mineral nutrients, organic nutrients, and plant growth regulators. Over small distances, substances move by diffusion and by cytoplasmic streaming, supplemented by active transport. Transport over longer distances proceeds through the vascular system, in bracket the xylem and the phloem, and is called translocation. An important aspect that needs to be considered is the direction of transport. In rooted plants, transport in xylem, in bracket of water and minerals, is essentially unidirectional from roots to the stems. Organic and mineral nutrients, however, undergo multidirectional transport. Organic compounds synthesized in the photosynthetic leaves are exported to all other parts of the plant, including storage organs. From the storage organs, they are later re-exported. The mineral nutrients are taken up by the roots and transported upwards into the stem, leaves and the growing regions. When any plant part undergoes senescence, nutrients may be withdrawn from such regions and moved to the growing parts. Hormones or plant growth regulators and other chemical stimuli are also transported, though in very small amounts, sometimes in a strictly polarized or unidirectional manner, from where they are synthesized to other parts. Hence, in a flowering plant, there is a complex traffic of compounds, in bracket but probably very orderly, moving in different directions, each organ receiving some substances and giving out some others. 11.1 Means of Transport. 11.1.1 Diffusion. Movement by diffusion is passive and may be from one part of the cell to the other, or from cell to cell, or over short distances, say from the intercellular spaces to the leaf to the outside. No energy expenditure takes place. In diffusion, molecules move in a random fashion, the net result being substance moving from regions of higher concentration to regions of lower concentration. Diffusion is a slow process and is not dependent on a living system. Diffusion is obvious in gases and liquids, but diffusion in solids rather than of solids is more likely. Diffusion is very important to plants since it is the only means of gaseous movement within the plant body. Diffusion rates are affected by the gradient of concentration, the permeability of the membrane separating them, temperature and pressure. 11.1.2 Facilitate diffusion. As pointed out earlier, a gradient must already be present for diffusion to occur. The diffusion rate depends on the size of the substances. Obviously, smaller substances diffuse faster. The diffusion of any substances across a membrane also depends on its solubility in lipids, the major constituent of the membrane. Substances soluble in lipids diffuse through the membrane faster. Substances that have a hydrophilic moiety find it difficult to pass through the membrane. Their movement has to be facilitated. Membrane proteins provide sites at which such molecules cross the membrane. They do not set up a concentration gradient. A concentration gradient must already be present for molecules to diffuse, even if facilitated by the proteins. This process is called facilitated diffusion. In facilitated diffusion, special proteins help move substances across membranes without expenditure of ATP energy. Facilitated diffusion cannot cause net transport of molecules from a low to a high concentration. This would require input of energy. Transport rate reaches a maximum when all of the protein transporters are being used, in bracket saturation. Facilitated diffusion is very specific. It allows cell to select substances for uptake. It is sensitive to inhibitors which react with protein side chains. The proteins form channels in the membrane for molecules to pass through. Some channels are always open, others can be controlled. Some are large, allowing a variety of molecules to cross. The porins are protein that form huge pores in the outer membranes of the plastids, mitochondria and some bacteria, allowing molecules up to the size of small proteins to pass through. Figure 11.1 shows an extracellular molecule bound to the transport protein. The transport protein then rotates and releases the molecule inside the cell, that is water channels made up of 8 different types of aquaporins. 11.1.2.1 Passive sympautes and antipodes. Some carrier or transport proteins allow diffusion only if 2 types of molecules move together. In a sympode, both molecules cross the membrane in the same direction. In an antipode, they move in opposite directions. In bracket, figure 11.2. When a molecule moves across a membrane independent of other molecules, the process is called unipode. 11.1.3 Active transport. Active transport uses energy to pump molecules against a concentration gradient. Active transport is carried out by membrane proteins. Hence, different proteins in the membrane play a major role in both active as well as passive transport. Pumps are proteins that use energy to carry substances across the cell membrane. These pumps can transport substances from a low concentration to a high concentration. In bracket, uphill transport. Transport rate reaches a maximum when all the protein transporters are being used or are saturated. Like enzymes, the carrier protein is very specific in what it carries across the membrane. These proteins are sensitive to inhibitors that react with protein side chains. 11.1.4 Comparison of different transport processes. Table 11.1 gives us a comparison of the different transport mechanisms. Proteins in the membrane are responsible for facilitated diffusion and active transport and hence show common characteristics of being highly selective. They are liable to saturate, respond to inhibitors and are under hormonal regulation. But diffusion, whether facilitated or not, takes place only along a gradient and do not use energy. 11.2 Plant-water relations. Water is essential for all physiological activities of the plant and plays a very important role in all living organisms. It provides the medium in which most substances are dissolved. The protoplasm of the cells is nothing but water in which different molecules are dissolved and in brackets several particles suspended. A watermelon has over 92% water. Most herbaceous plants have only about 10 to 15% of its fresh weight as dry matter. Of course, distribution of water within a plant varies. Woody parts have relatively very little water, while soft parts mostly contain water. A seed may appear dry, but it still has water, otherwise it would not be alive and respiring. Terrestrial plants take up huge amount of water daily, but most of it is lost to the air through evaporation from the leaves, that is transpiration. A mature corn plant absorbs almost 3 liters of water in a day, while a mustard plant absorbs water equal to its own weight in about 5 hours. Because of this high demand for water, it is not surprising that water is often the limiting factor for plant growth and productivity in both agricultural and natural environments. 11.2.1 Water potential. To comprehend plant-water relations, an understanding of certain standard terms is necessary. Water potential in bracket psi W is a concept fundamental to understanding water movement. Solute potential in bracket psi S and pressure potential in bracket psi P are the two main components that determine water potential. Water molecules possess kinetic energy. In liquid and gaseous form, they are in random motion, that is both rapid and constant. The greater the concentration of water in a system, the greater is its kinetic energy or water potential. Hence, it is obvious that pure water will have the greatest water potential. If two systems containing water are in contact, random movement of water molecules will result in net movement of water molecules from the system with higher energy to the one with lower energy. This water will move from the system containing water at higher water potential to the one having low water potential. This process of movement of substances down a gradient of free energy is called diffusion. Water potential is denoted by the Greek symbol psi or psi and is expressed in pressure units such as pascals. By convention, the water potential of pure water at standard temperatures, which is not under any pressure, is taken to be 0. If some solute is dissolved in pure water, the solution has fewer free water and the concentration of water decreases, reducing its water potential. Hence, all solutions have a lower water potential than pure water. The magnitude of this lowering due to dissolution of a solute is called solute potential or psi S. Psi S is always negative. The more the solute molecules, the lower, in bracket more negative, is the psi S. For a solution at atmospheric pressure, in bracket water potential, psi W is equals to psi S, in bracket solute potential. If a pressure greater than atmospheric pressure is applied to pure water or a solution, its water potential increases. It is equivalent to pumping water from one place to another. Can you think of any system in our body where pressure is built up? Pressure can be built up in a plant system when water enters a plant cell due to diffusion causing a pressure built up against cell wall. It makes the cell turgid. In bracket see section 11.2.2. This increases the pressure potential. Pressure potential is usually positive, though in plants negative potential or tension in the water column in the xylem plays a major role in water transport of a stem. Pressure potential is denoted as psi P. Water potential of a cell is affected by both solute and pressure potential. The relationship between them is as follows. Psi W is equals to psi S plus psi P. 11.2.2 Osmosis. The plant cell is surrounded by a cell membrane and a cell wall. The cell wall is freely permeable to water and substances in solution, hence is not a barrier to movement. In plants, the cell usually contain a large central vacuole whose contents the vacuolar shape contribute to the solute potential of the cell. In plant cell, the cell membrane and the membrane of the vacuole, the tonoplast together are important determinants of movement of molecules in or out of the cell. Osmosis is a term used to refer specifically to the diffusion of water across a differentially or semi-permeable membrane. Osmosis occurs spontaneously in response to a driving force. The net direction and rate of osmosis depends on both the pressure gradient and concentration gradient. Water will move from its region of higher chemical potential in bracket or concentration to its region of lower chemical potential until equilibrium is reached. At equilibrium, the two chambers should have the same water potential. You may have made a potato osmometer at some earlier stage in school. If the tuber is placed in water, the cavity in the potato tuber containing a concentrated solution of sugar collects water due to osmosis. Study figure 11.3 in which the two chambers A and B containing solutions are separated by a semi-permeable membrane. A. Solution of which chamber has a lower water potential. B. Solution of which chamber has a lower solute potential. C. In which direction will osmosis occur? D. Which solution has a higher solute potential. E. At equilibrium, which chamber will have lower water potential. And F. If one chamber has a psi of 2000 kilopascal and the other 1000 kilopascal, which is the chamber that has the higher psi. Let us discuss another experiment where a solution of sucrose in water taken in a funnel is separated from pure water in a beaker through a semi-permeable membrane. In bracket figure 11.4. You can get this kind of membrane in an egg. Remove the yolk and albumin through a small hole at one end of the egg and place the shell in dilute solution of hydrochloric acid for a few hours. The eggshell dissolves, leaving the membrane intact. Water will move into the funnel, resulting in rise in the level of the solution in the funnel. This will continue till the equilibrium is reached. In case sucrose does diffuse out through the membrane, will this equilibrium be ever reached? External pressure can be applied from the upper part of the funnel such that no water diffuses into the funnel through the membrane. This pressure required to prevent water from diffusing is in fact the osmotic pressure, and this is the function of the solute concentration. More the solute concentration, greater will be the pressure required to prevent water from diffusing in. Numerically, osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. Osmotic pressure is the positive pressure applied, while osmotic potential is negative. 11.2.3 Plasmolysis. The behavior of the plant cell in bracket or tissues with regard to water movement depends on the surrounding solution. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. If the external solution is more dilute than the cytoplasm, it is hypotonic, and if the external solution is more concentrated, it is hypertonic. Cells swell in hypotonic solutions and shrink in hypertonic ones. Plasmolysis occurs when the moves out of the cell and the cell membrane of a plant cell shrinks away from its cell wall. This occurs when the cell in bracket or tissues is sprayed in a solution that is hypertonic, in bracket has more solutes, to the protoplasm. Water moves out, it is first lost from the cytoplasm and then from the vacuole. The water when drawn out of the cell through diffusion into the extracellular, in bracket outside cell, fluid causes the protoplast to shrink away from the walls. The cell is said to be plasmolysed. The movement of water occurred across the membrane, moving from an area of higher water potential, that is the cell, to an area of lower water potential outside the cell. In bracket figure 11.5. What occupies the space between the cell wall and the shrunken protoplast in the plasmolysed cell? When the cell in bracket or tissue is placed in an isotonic solution, there is no net flow of water towards the inside or outside. If the external solution balances the osmotic pressure of the cytoplasm, it is said to be isotonic. When water flows into the cell and out of the cell and are in equilibrium, the cells are said to be flaccid. The process of plasmolysis is usually reversible. When the cells are placed in a hypotonic solution, in bracket higher water potential or dilute solution as compared to the cytoplasm, water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall. That is called turgor pressure. The pressure exerted by the protoplast due to entry of water against the rigid cell wall is called pressure potential, psi p. Because of the rigidity of the cell wall, the cell does not rupture. This turgor pressure is ultimately responsible for enlargement and extension growth of cells. What would be the psi p of a flaccid cell? Which organisms other than plants possess cell wall? 11.2.4 Imbibition. Imbibition is a special type of diffusion when water is absorbed by solids, colloids, causing them to enormously increase in volume. The classical examples of imbibition are absorption of water by seeds and dry wood. The pressure that is produced by the swelling of wood had been used by prehistoric men to split rocks and boulders. If it were not for the pressure due to imbibition, seedlings would not have been able to emerge out of the soil into the open. They probably would not have been able to establish. Imbibition is also diffusion, since water movement is along a concentration gradient. The seeds and other such materials have almost no water, hence they absorb water easily. Water potential gradient between the absorbent and the liquid imbibed is essential for imbibition. In addition, for any substance, the imbibe any liquid, affinity between the adsorbent and the liquid is also a prerequisite.